Hello, everyone, and welcome to Genome Webinars. I'm Ben Butkus, Editorial Director for Genome Web, and I'll be your moderator today. Today's webinar is entitled Implementation of Digital PCR in a Diagnostic Lab, Evaluation of Minimal Residual Disease. The sponsor of this webinar is Stilla Technologies. Our presenter today is Benjamin Tournier of the Molecular Pathology Department at University Hospital Dijon. A reminder to attendees, you may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you do not see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentation has concluded. We'll now begin the webinar. Please go ahead, Benjamin.
Thank you, Ben. And so hi, everybody. Firstly, I would like to thank Genome Web for giving me the opportunity.
Excuse me, Benjamin?
Yes.
One moment, please. Can you adjust your headset? We're having some audio difficulties.
Thank you, Ben. And firstly, I would like to
Please, Benjamin, please hold on. Stand by one moment, please.
Okay. So, hi everybody. Thank you, Ben, for your introduction. Firstly, I would like to thank Genome Web for giving me the opportunity to present our experience in the platform of semantic for giving me the opportunity to present our experience in the semantic platform of oncology in Dijon. And I will present you the digital PCR system that we use in Dijon, developed by Stilla Technologies. So maybe you are not familiar with Dijon. It's the capital city of the region called Bourgogne, which is a famous region of France, known for its wine and mustard. So just after this funny introduction, I will focus on the main topic of this presentation, which is the liquid biopsy. The concept of liquid biopsy is rather recent and consists of the exploration of biomarker in the blood of patients. And this illustration shows you the principal application in the field of oncology for the detection and characterization of circulating tumor DNA, circulating tumor cells, and also circulating tumor exosomes. So in the first part of my presentation, I will show you how we set up the liquid biopsy process in our platform. So here is just a slide to show you numbers about our platform. This platform was created 10 years ago. We performed more than 2,000 analyses of solid tumor per year. And since 2016, we also perform liquid biopsy analysis, mainly for melanoma and lung cancer patients. And for example, in 2017, we performed liquid biopsy analysis for melanoma patients, mainly in the context of follow-up, whereas for lung cancer patients, it was mainly in the context of diagnosis and progression. So you have to know that the point that is very important for the liquid biopsy, firstly, is the choice of the collection tube. So we choose two different types of tubes depending on where is performed the blood sampling. For blood sampling that are performed in our hospital, we use the Key2 EDTA tubes. And for blood sampling that are performed far from our hospital, we use cell-free DNA collection tube from Roche. You have to know that there are several other commercial tubes available, and you see here some examples. We did not test all of them, but we know that we obtain good results with Roche tubes. And you have here two recent publications comparing some of them, including the tube from Roche. Here is a representation of our liquid biopsy process. So the time between the blood sampling and the preservation at -80 degrees is very important. For EDTA tubes, we have to limit this time to 4 hours, and sometimes, and most of the times, it's about 1 or 2 hours. And for cell-free DNA collection tube, we have to limit to 7 days, but also for these tubes, it's about 5 days. So we perform 2 centrifugation. The first is to isolate the plasma. As I said before, just after this isolation, we put some plasma samples at -80 degrees. Then, before the analysis of plasma, we bow plasma by putting them at 4 degrees 1/9, and after 1 hour at room temperature, then we perform the second centrifugation, and we perform the cell-free DNA extraction and molecular analysis. So you also have to know that the pre-analytical step is very crucial for the integrity of cell-free DNA. That's why we respect this strict process. Concerning the DNA extraction method, we use 2 methods. First, an automated extraction. Most of our samples are extracted using this instrument, which is a Kia Symphony. So we started with 4 ml of plasma, and the elution is in 60 microliters. In some cases, we have also a manual extraction using the kit from Maschrenagel. And we use this method when the quantity of plasma is not sufficient, and for some other fluid, like TSF, urine. We can extract up to 5 ml of plasma, and the elution is in 45 microliters. And also, this extraction method is our backup extraction method. At the end of this process, it's important to quantify cell-free DNA and to qualify it. To quantify, we use the qubit DNA high sensitivity. And for the qualification of the DNA fragment, we use the tape station. Here is an electrophoregram showing you a classical profile of cell-free DNA. Most of the content of cell-free DNA is above 100 base pair and up to 300 base pair. We also look at the gDNA contaminant by looking at fragments that are above 500 base pair. And when there are too many long fragments, we can conclude sometimes that there is a contamination. So it's particularly useful to evaluate if the sample is interpretable. To conclude with the pre-analytical steps, it's very important to extract a minimal volume of 4 ml of plasma. Also, the transport time is very crucial for to maintain the integrity of cell-free DNA. We also perform a double centrifugation, particularly to avoid the extraction of cells present in plasma. And finally, we respect a gentle vowing, also in order to limit the degradation of the sample. So now, in the second part of the presentation, I will show you the Naica Digital PCR system. So the principle of this system is based on the use of a chip called Sapphire. So it's this chip, which is already loaded with oil. So we only have to load PCR reagent and cell-free DNA. This chip is composed of 4 chambers. And here you have an illustration of the underside of this chip. So we can analyze 4 samples per chip. And here is an enlargement of a chamber when you see the microfluidic channels for the distribution of the mixture. And it is possible to read fluorescence in 3 different fluorescence channels. This system is composed of 3 instruments that take little space. First, an air pump, which provides air pressure for the second instrument, which is called Naica Geode. This instrument performs the droplet generation and the thermocycling. And finally, the third instrument is called Naica Prism 3 and performs the fluorescence readout. This slide shows you the generation and of droplets and the site calibration. So as you see, the generation of droplets is made by passing through injectors, and after the size of droplets is calibrated thanks to a grid present in the chamber. And here is a photography of the results that are obtained at the end of the thermocycling. So here is a photograph of a chamber in the blue channel after the thermocycling. You see each droplet. And you see also that each white spot represents a droplet in which we observe a signal, a positive fluorescent signal. As you see, the distribution of droplets is here pretty good. And thanks to this representation, it is possible to check the global quality of the results, and particularly to exclude some events that I will show you after. This previous representation can be then transformed into a one-dimensional chart that you see here. The number of generated droplets is here above 26,000, which is good, and there is very little range of droplets between positive droplets and negative droplets. So as I said before, it is possible to check the quality of the result. So you have here an example of artifact. And so it's a photography here in the green channel. We do not observe positive droplets, except in this area in which we observe a big area, a big white artifact. And you have here an enlargement. You see that it do not correspond to droplets. So if we look at the one-dimensional chart, we observe for this example some positive droplets, which in fact correspond to this artifact. So by excluding this area, thanks to the software, we no longer observe these false positive events. So by this way, it's a means to control the quality and to exclude potential false positive results. You have to know that the nature of the samples can influence also the fluorescence intensity, particularly depending on the extraction method and the buffer that are used. In these examples, you see that the level of fluorescence for positive droplets is different for these 5 samples. If we set up a same threshold of fluorescence, some of the positive droplets are missing. So it's also possible to adapt the threshold individually in each chamber. And so by doing that, we can analyze all positive droplets. So now in the third part of my presentation, I will show you some example of application. And the first is for the BRAF testing in melanoma. So in this context, we use a design targeting the most common BRAF mutation in melanoma. So you see here this design. This design uses 3 hydrolysis probes. The first is a white-type probe labeled in blue. A second probe labeled in green targeting the V600K mutation, and the V600E mutation probe labeled in red. So here is the step that we followed for the validation of all our molecular tests, including the BRAF test. So first, a classical PCR optimization, then an evaluation of the PCR efficiency by qPCR, and finally, an evaluation of the signal noise in order to determine a limit of detection above which a sample is considered as positive. For example, for the BRAF test, we analyze 25 cell-free DNA samples without BRAF mutation. In this example, we observed in the green channel 1 positive droplet in only 3 samples. So we set a limit of detection of 6 droplets for this channel. We followed the same strategy for the red channel, and we also set a limit of detection of 6 droplets in this example. So now I will show you an example of follow-up by liquid biopsy for a patient who was diagnosed for a melanoma in 2008. And this patient displayed a mutation of BRAF in his preliminary tumor. So we started the follow-up by liquid biopsy in order to determine the efficiency of the immunotherapy in 2016. So in this graph, you see that the green area indicates the concentration of the cell-free DNA, and the red line indicates the concentration of the BRAF mutation in plasma. And of course, these values were determined using digital PCR. During the first 8 months of the follow-up, these values were stable, coinciding with the stable F status of the patient. Then we detected the BRAF mutation in 2 liquid biopsy, just above our limit of detection. Unfortunately, we did not confirm this result due to missing points in the next month. That's why after we observed a strong increase of the mutation in the plasma. This was an argument for clinicians to quickly change the therapy by an anti-BRAF and anti-MEK therapy. So just after, we observed that the mutation dropped, and at the opposite, we observed a strong increase of the cell-free DNA concentration. And this was observed with the treatment response. And finally, we observed again an increase of the BRAF mutation in plasma, followed by the progression of the cancer, and finally the death of the patient. So here is an example, was our first example of long-time survival of patients, and it helped us to improve our follow-up by liquid biopsy. Finally, I will finish my presentation by show you the BRAF testing, the EGFR testing in the lung cancer. So here it's an example of design that we use for lung cancer. And this design is dedicated to the detection of the activating mutation of EGFR, so the mutation L858R mutation, thanks to an hydrolysis probe labeled in red, and also for the detection of the exon 19 of EGFR. For this deletion, in fact, we use the combination of 2 hydrolysis probes. A first probe labeled in blue hybridized in a region where no deletion occurs, whereas the probe labeled in green hybridized in a region which is a hotspot deletion region. So for a sample which is wild type, you have the hybridization of the 2 probes. But when a sample displays a deletion, only the blue probe hybridized. So here is an example in one-dimensional chart for a sample which was analyzed for EGFR. And you see that in this example, it's not possible to identify the deletion, but by using 2-dimensional charts, combining the green channel and the blue channel, we can visualize the deletion. You have here a big cloud of double positive droplets for green and blue channels, which correspond to the hybridization of the two probes. And you have here a small cloud of monopositive droplets, so blue monopositive droplets, which correspond to the copies of DNA displaying the deletion. So we observed 48 monopositive droplets, which correspond in this case of 5 mutated copies per microliter. Here is the same representation, but for DNA without deletion of EGFR. As you see, we do not observe a clean cloud of monopositive droplets. We only observed 5 monopositive droplets, and which is above our limit of detection for this test. So in this example, we consider this sample as negative. Here is a second example of design for the detection of the activating mutation of EGFR L858R mutation, L861Q mutation, and the resistance mutation T790M mutation. So it's here a rather simple design. You have a probe labeled in red for the L858R mutation. Again, a probe labeled in red for the V861Q mutation, and the white type probe labeled in blue. For the resistance mutation, the probe is labeled in green. And now I will show you an example of progression for a lung cancer patient using the design that I showed you previously. So this patient was diagnosed for a lung metastatic adenocarcinoma in 2014. We detected the activating mutation of EGFR on a tissue biopsy, and so this patient was treated by an anti-EGFR therapy. We firstly analyzed a liquid biopsy in February 2017. In this liquid biopsy, we detected the initial activating mutation. So you have here the blue channel corresponding to the white type probe, the green channel corresponding to the resistant mutation, and the red channel corresponding to the activating mutation. You have here the results for the sample, here for the white type, again here sample white type. And you see here the droplets for the activating mutation. So in this example, 6 droplets is just above our limit of detection, so we considered this example as mutated. But we only observed 1 positive droplet for the resistance mutation, so we considered as negative for the resistance mutation. After, in March 2017, we analyzed the biopsy from vertebral metastasis, but we did not detect any mutation, possibly due to a lack of tissue. So that's why we performed a second liquid biopsy in July 2017, and this time we observed a strong increase of L858R mutation. You see here 551 positive droplets, which is very important. And we also observed positive droplets for the resistant mutation. So now it was an argument for clinicians to change the previous anti-EGFR therapy by a third generation anti-EGFR therapy targeting the resistance mutation. So now I will conclude my presentation by a slide showing you our strategy of digital PCR depending on the context of the disease. So as you see, for melanoma, in the context of diagnostic or follow-up, we perform the testing of BRAF, but in the context of diagnostic, we also perform high-throughput sequencing if we do not detect BRAF mutation. In order to detect mutation in other gene implicated in the MAP kinase pathway. For lung cancer, in the context of diagnostic, we perform the analysis of EGFR deletion of exon 19 and activating mutation L858R mutation. And again, if we do not detect mutation, we do NGS analysis. And for the progression, we have two possibilities depending on the initial activating mutation. So either the L858R mutation, L861Q mutation, or the deletion in combination with the resistance mutation. If we do not detect this mutation, we also perform an analysis of the MET gene in order to determine a possible amplification of this gene. Sorry. And finally, I would say that about this system, that it's really a fast workflow that gives us the possibility to give results in less than 1 day, from the blood sampling to the digital PCR results, which is pretty good for our clinicians. It's also possible to multiplex thanks to 3 fluorescence channels. So it's particularly adapted for the diagnosis of several mutations. And finally, this technique is very sensitive and reliable, which is adapted to the follow-up of patients for us. So here are all my collaborators, and I thank you for your attention. So now, Ben, it's up to you.
Thank you, Benjamin. A reminder to attendees, you may type in a question at any time. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you do not see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We'll now begin the Q&A portion of the webinar. The first question: why don't you use double spin before freezing as recommended by many authors?
Okay. Yes, we perform the double centrifugation. The first is to isolate, of course, the plasma, and the second centrifugation is just after VAU samples, because sometimes some protein precipitates, and including maybe some exosomes and some cells, some residual cells, and so it's to avoid the contamination of either genomic DNA or exosome containing DNA or RNA. So it's really to be sure to analyze cell-free DNA.
Thank you. The next question, are your probes and primers custom designed?
Yes, we design all of our primer and probes, except for the first design of EGFR and BRAF. Styla Technologies provide us sequence of probes and primers to begin our analysis, and this sequence are available in my presentation. You have the link of the publication, so you can retrieve the sequence if you want.
Thank you. Next question. Did you test the Biorad QX200 system as well as the Naica system? And if so, why did you choose the Naica system?
Yes, in fact, before purchasing the system from Naica, we test also the system from Biorad. In fact, we test the analysis of samples using the system from Biorad and using the Naica system. We obtained the same results in terms of sensitivity, and so it was the same for us. But with the system from Naica, from Styla, we thought that the workflow was more simple, and also there was the possibility to multiplex. So that's why we choose the system from Styla. And also, yeah, it was all the workflow was fast. So it was an element to choose the system from Styla.
Thank you. The next question. How much DNA, how many picograms or nanograms do you use in 14 microliters?
In fact, it's depending on the samples, because in fact we analyze we analyze all of it's possible to analyze, in fact, and sometimes in most of the cases we obtain maybe 1 nanogram per microliter after extraction, maybe less, and we analyze so 14 microliter in all of the cases. So it's dependent of the concentration. Sometimes it's 40 nanogram, maybe sometimes it's more. We are not limited with the quantity. In fact, it's rather rare to have a case of saturating the system with a lot of DNA quantity. Most of the time we are in the case where we have not enough DNA, so it's not a limited criteria.
Thank you. The next question. How many wells are there in each digital PCR chamber?
I don't know if if the question is really good, because there are no wells in the chamber. In fact, you have a chip that consists of 4 chambers for the analysis of 4 samples. In each of these chambers, it is possible to generate droplets, which is about 25 to 30,000 droplets. So it's variable, but in most of the times we obtained more than 25,000 droplets. So you have 25,000 droplets per samples.
Thank you. The next question. How do you set up the threshold for calling a signal positive or negative? For example, in some of the figures you showed, with a minimal change of the threshold line, the number of positive signals could change.
So we perform also analysis of, as I showed you previously, we perform the analysis of wild-type cell-free DNA samples extracted with the same method in order to limit the variability of fluorescent threshold. And we set up a threshold that allow us to include a maximum number of positive droplets and also a minimal number of positive droplets in samples displaying no DNA. And yes, that's how we set up the threshold. And yeah, sometimes we observe some samples with, as I showed you previously, with fluorescence intensity which is different. And for these cases, we adapted the threshold for these conditions.
Thank you. The next question. Did you compare digital PCR with a proximity ligation assay? If yes, which technology was more sensitive?
No, we did not compare we did not compare digital PCR with proximity ligation assay. I'm sorry, we did not perform this comparison of technology.
Thank you. The next question. Does the Naica chip contain oil, or does it have to be inserted?
No, as I said before, these chips are already loaded with oil, so we only have to we prepared the PCR with the PCR reagent and the cell-free DNA, and after we put it directly on the chip. It's rather simple. We don't have to load oil.
Thank you. The next question. Is it correct to assume that for solid tumors, the initial diagnosis is performed on tissue while the monitoring or progression is monitored through liquid biopsy?
In fact, it is dependent of the authorization of it's dependent of the cancer and of the law. For example, in lung cancer, and it's always preferable to do the analysis on solid tumors, but sometimes where it's not possible to analyze the solid tumor, it's possible to give a result using the liquid biopsy. So in this type of cases, the liquid biopsy can substitute the tumor tissue, But for other cancer, for now, there are no arguments to indicate that it is preferable to use liquid biopsy. So in other cancers, it's preferable to use liquid biopsy only for the follow-up. But it's always preferable to look at the tumor to be sure of the nature of the cancer.
Thank you. The next question. What is the limit of detection for digital PCR on the Naica?
And the limit of detection for the system Naica, firstly, is dependent of our assay. And but anyway, with this system, we have a limit of detection of 0.2 copies per microliter. But of course, after you have to consider the limit of detection of your assay, depending of the design.
Thank you. The next question. Do you have to use specific digital PCR master mix reagents?
Yes. So for now, there is one PCR reagent which is recommended by Stila, and which is the Quanta Perfecta fourth mix, which is very good, and you can use also for qPCR. And you can, if you want, test other mix for PCR. And if you want to test it, to validate it, it's possible. And but for Stila, only the mix for Perfecta is optimized. But it's always possible to test other mix if it works in your conditions. And yes, I just would add that yes, it's an open system, and sometimes the first criteria that is important is it's to control that your mix do not disturb the generation of droplets. So you can use several other reagents to adapt the viscosity in order to be sure that the generation of droplets is good.
Thank you. The next question. How many samples can you process at a time using the Naica?
So the PCR, the Sapphire chip consists of 4 chambers, and it is possible to perform the thermocycling of 3 chips per run. So it is possible to analyze per run 12 samples, and after the time of the run, so the time of the PCR thermocycling and generation of droplets, it's about 2 hours, 2 or 3 hours, depending on your program of PCR. So after, in a day, you can perform several runs of digital PCR, up to maybe 4 runs. So if you optimize your workflow, it is possible to analyze 48 samples.
Thank you. The next question, how long does it take from droplet generation to data with the Naica system?
So, firstly, you have to prepare your PCR mixture. So It's depending on the number of samples you have to prepare, and we can evaluate this time for 1 hour to maybe 10 minutes. After, you have to load the chip, which is if you consider a free chip, it's maybe 5 to 10 minutes, depend of the technician. After, you have the PCR thermocycling, and I said before that is dependent on your program, but normally it's about 2 to 3 hours. So if I consider, and after for the analysis, it's rather fast. I would say 15 minutes. So if I include all these times, it's about 4 hours. No, 3 hours.
Thank you. The next question. Have you looked at whether gentle thawing improves cell-free DNA recovery?
In fact, it's rather a recommendation. We did not test a comparative. We did not perform a comparative study of the time of thawing, but we know that it's preferable to do this, and we do not have the results for now to show you that is preferable.
Thank you. The next question. What is the minimal DNA fragment size that can be used for the reaction? Do you need to check the size beforehand using a bioanalyzer, for instance?
Yeah. Again, it's dependent of your design and of the size of your amplicon. But most of the time, the size of cell-free DNA is above 120, 130 base pair. So if you design an amplicon above 80 base pair, it's good for all of the cases. But in some rare cases, we encounter cases with a degradation more important of cell-free DNA. That's why in some cases it's important to control the size in order to be sure that that if you do not observe positive droplets for the wild type DNA in your PCR, to control that it is not due to not enough material, but that is not due to degradation of DNA. So yeah, it's dependent of your assay, but you always have to control the size of the DNA by tip station or comparable techniques.
Thank you. The next question, approximately how many liquid biopsy samples do you analyze per year?
I have to evaluate. We have not a stable quantity of liquid biopsy that we analyze per week. I would say maybe we analyze six liquid biopsy per week. So if we do the multiplication, and it's it's 312. It's about 312 analyses per year, but it's not stable for now because each year we observe an increase of the number of liquid biopsies. So for now it's rather this number, but maybe next year it will be up.
Thank you. The next question. Have you confirmed positive results using next-generation sequencing?
Yes and no. Sometimes, most of the times it's the opposite. We use a digital PCR to confirm a variant that we detect in NGS, because for now we do not have the same sensitivity, and but when we detect mutation in NGS, either it's frequent that is really important and we are sure of this mutation, and when it is not the case, either we confirm by a second NGS analysis, or we send the sample in our platform using NGS technologies which are more sensitive.
Thank you. This concludes the Q&A portion of the webinar. We'd like to thank our sponsor, Stilla Technologies, and our presenter, Benjamin Tournier, of the Molecular Pathology Department at University Hospital, Dijon. If we didn't have time to get to your questions, we will try to have our panelists answer them directly afterward. As a reminder, please look out for the pop-up survey from the sponsor after you log out. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to attendees. Thank you for joining us for this Genome webinar.